And if you miss a class, you can have access to the lecture. Or if you're going through your notes and there's something that doesn't make sense, or you had one of these situations where you're taking notes and you start to drift off a little bit, you can go back to the lecture and kind of pick up where you missed something. Okay, so those will be posted routinely, and just another resource that I wanted you to be aware of. So any questions so far? Okay, a little bit about how this class will differ from Biology 134. And as an introduction to that, I'd like to play you a little 3-minute video clip by Richard Feynman. Has anybody ever heard of Richard Feynman? Anybody watch Neil deGrasse Tyson at all on TV? He talks about Feynman all the time. Anybody watch The Big Bang Theory? They talk about Feynman quite a bit. Feynman's a physicist, and a very famous physicist. And he, in this little video, makes an analogy between kind of the process of science and a cosmic chess game. So let's take a look and see what Feynman has to say about science and cosmic chess. If I plugged in this.
That the gods are playing some great game like chess.
Let's start this over again.
One way, it's kind of a fun analogy to try to get some idea of what we're doing and try to understand nature, is to imagine that the gods are playing some great game like chess, let's say a chess game, and you don't know the rules of the game, but you're allowed to look at the board, at least from time to time, and in a little corner perhaps. And from these observations, you try to figure out what the rules are of the game, what the rules of the pieces mean. You might discover after a bit, for example, that when there's only one bishop around on the board, that the bishop maintains its color. Later on, you might discover the law for the bishop as it moves on a diagonal, which would explain the law that you understood before, that it maintains its color. And that would be analogous to we discover one law and then later find a deeper understanding of it. Then things can happen. Everything's going good. You got all the laws. It looks very good. And then all of a sudden, some strange phenomenon occurs in some corner. So you begin to investigate that, to look for it. It's castling, something you didn't expect it. We're always, by the way, in a fundamental physics, always trying to investigate those things in which we don't understand the conclusions. We're not trying to check all the time our conclusions. After we check them enough, we're okay. The thing that doesn't fit is the thing that's the most interesting. The part that doesn't go according to what you expected. Also, we could have revolutions in physics after you've noticed that the bishops maintain their color and they go along the diagonals and so on for such a long time, and everybody knows that that's true. Then you suddenly discover one day in some chess game that the bishop doesn't maintain its color. It changes its color. Only later do you discover a new possibility, that the bishop is captured and that a pawn went all the way down to the queen's end to produce a new bishop. That can happen when you didn't know it. And so it's a very analogous to the way our laws are. They sometimes look positive. They keep on working, and all of a sudden some little gimmick shows that they're wrong. And then we have to investigate the conditions under which this bishop change of color happened. And so and gradually learn the new rule that explains it more deeply. Unlike the chess game, though, in the case of the chess game, the rules become more complicated as you go along. But in the physics, when you discover new things, it looks more simple. It appears on the whole to be more complicated because we learn about a greater experience. That is, we learn about more particles and new things. And so the laws look complicated again. But if you realize all the time, it's kind of wonderful. That as we expand our experience into wilder and wilder regions of experience, every once in a while we have these integrations in which everything is pulled together in a unification, which it turns out to be simpler than it looked before.
The analogy is kind of this. The study of science and the investigations into life and living phenomena are kind of like trying to learn the rules of a game, in this particular case chess, without having the instruction book. And the way that you can do that is by observing. Observation is critical in the scientific method. Developing hypotheses, testing those hypotheses, validating the data from your hypothesis are all important parts of this phenomena of deductive reasoning. And so we learn a whole lot by observing and trying to generalize and make these rules or laws or theories of science. One of the things that really helps us in our observation is technology. And one of the things we're going to do this semester is we're going to use some technology that allows us to try to extend our normal senses to be able to detect things that we couldn't otherwise see. Okay, we can measure in wavelengths, for example, that the human eye has difficulty seeing using a spectrophotometer. And we can quantitate those wavelengths and put some numbers associated with various absorbance readings. So the first semester, 134, was more about process. And it was about learning how scientists think, about designing experiments, making observations. And we really weren't that content driven in the first semester of the course. This course shifts emphasis now a little bit. And basically what we're going to be doing is we're going to be looking at what are some of the cornerstone ideas, concepts, laws, theories that have been developed through this scientific method, through this process of trying to learn the rules of the game without having the rule book, by making observations. And so we're going to talk about some of these cornerstone ideas, the cell theory, for example, the central dogma. And these are things that we have determined in this particular curriculum at Franklin College are going to be cornerstone concepts that you'll need in a number of additional courses that you'll take. It's very difficult to understand how a kidney functions, for example, in zoology or anatomy and physiology, if you'd understand the basics of osmosis and diffusion. It's very difficult to understand some of the molecular genetics that Dr. Morton McCombs will be doing in the genetics or the biotechnology class if you don't understand the central dogma. Okay? So this particular class is going to concentrate on kind of helping you to understand those particular important central concepts. And one of the things I like to do whenever possible is give you a little bit of background into how those particular ideas evolved and what kind of information people gathered to result in those particular generalities. And so some history of science creeps its way in to this course as well. Some students tend to find this course material, even though it's probably a little bit more detailed, a little bit easier to deal with and wrap their head around because it's not quite as esoteric as the first semester where you're talking more about kind of process of science. And so this is more kind of traditionally what you would expect from a biology class. We're going to learn about cell respiration and we're going to dig into the details of cell respiration. One of the things that is important to understand is it's important to keep up because concepts build on one another. And so if you find yourself struggling with some material and you've never quite mastered it, it's going to come back to bite you a little bit in future exams. And so if you're having difficulty with stuff, make sure you come in and get some help. You can get help from Austin in the mentoring sessions, but I'm available. I've got an open door policy. If you need help, come in and get help from me. If you need help with lab information, come and talk to Dr. Morton. Probably the easiest way to find us, if we're not just in our offices waiting to see you, is to talk to Natalie, because Natalie has our schedules and Natalie knows what we're doing. And if you want to make an appointment with us, it's easiest to come down to her office. She can pull up our schedules and then she can look at your schedule and say, okay, let's set up time. So we're going to go ahead and get started with some actual material today. So if you have paper and writing instruments, please pull them out. What I would strongly recommend that you do for this class is that you print out the PowerPoint slides as thumbnails. And that way you can go ahead and use those PowerPoint slides as a way to take your notes for the class. And by the way, when it comes to exams, the exams are very lecture-driven. The material that we will cover on the exams will be material we talk about in class. If there is something in the textbook that I have not specifically talked about in class or told you you are responsible for, then that is kind of just background information, but it's not something you're going to be evaluated on those lecture exams. So take good lecture notes, okay? That's going to be really important. So, what's the thing that probably jumps out at you most significantly about this view of Earth? If you were an alien, cruising into our solar system, and you saw this particular planet in front of you, what would strike you? Pardon me?
There's a lot of blue.
There's a lot of blue, okay? There's a lot of water. So why is water so important? Anybody follow any of the kind of explorations that NASA's still doing, looking for the possibility of life on other planets? Looks like Mars rovers and things, yeah.
It's extraordinarily rare to find a planet with water.
It's extraordinarily rare to find a planet with water, but we are starting to find more and more places where water exists that we didn't think it did. Why is NASA so hung up about water?
It helps support life.
Yeah, I mean, life as we know it, and that's a big caveat, life as we know it, cannot exist without water. And the idea being is, if there's not water present, then life as we know it probably does not exist. Okay? Why is water so important? Why is life so water dependent? Well, there's several attributes of water that are incredibly important. And some of those properties of water that life depend upon include things like the property of cohesion. What's cohesion? If a group is cohesive, what does it do? It works well together, it sticks together. Okay? Cohesion is the property of like molecules sticking to like molecules. Does anybody know where cohesion is a very important phenomena in a living system? Cohesion is a very important phenomena in a concept called transpirational pull. And transpirational pull explains how water can get from the soil to the top of a large plant, like this tree. There are water conducting cells in the trunk of a tree. Somebody know what those water conducting cells are called? They're actually dead cells. They're hollowed out cells. They make up the wood of the tree. Anybody ever heard of xylem? X-Y-L-E-M? Water moves through the xylem because of evaporation occurring at the leaves. And because water evaporates from the leaves, as one water molecule evaporates, another is pulled up behind it to replace it. Why? Because water molecules are cohesive. They're sticky. They tend to form a continuous column. Water also has a high heat of vaporization. Does anybody know what that refers to? Okay. It takes a lot of energy to change the temperature of water and to change its state from a liquid to a gas. High heat of vaporization is the basis behind evaporative cooling. And evaporative cooling, obviously, is an essential element of the function of a multicellular organism like us, sweating. If our body heats up too much, then bad things start happening within your cells. Specifically, proteins start to denature, and you start losing enzyme function. High specific heat is another property of water, and that again refers to the fact that it takes a lot of energy to change the temperature of water. Because water has a high specific heat, water can effectively act as a buffer and mitigate temperature changes. That's one of the reasons why the climate of our planet is pretty hospitable, because of the large amounts of water that are present. What happens when we have a lot of solar gain during the daytime? Water absorbs that heat, and it takes a lot of energy to appreciably change the temperature of that water. At nighttime, when the sun stops shining, the heat that the water is stored is slowly re-radiated back from the atmosphere again, and that mitigates temperature changes of the atmosphere. And so water helps to moderate the temperature of our planet and leave it in that quote-unquote Goldilocks zone where the temperature of the planet is just right for organisms that are living there. Water is less dense as a solid than a liquid. That's an unusual property. Can anybody think of an example where that might be important for water to be less dense as a solid than a liquid?
Fish.
For fish, and specifically the fact that ice floats, right? Because ice floats, that ice that floats to the top of a body of water creates an insulating layer and helps to prevent bodies of water that aren't too shallow from completely freezing all the way down. If water didn't have this particular property, bodies of water would freeze from the bottom up. And that wouldn't be very conducive to organisms maintaining viability during the winter in aquatic environments. Water is sometimes called the universal solvent. What's an exception to water being a universal solvent? What kind of things don't dissolve in water? A lot of organic compounds that are hydrocarbons. The lipid molecules are notoriously water insoluble. Under what situations in your body is it important that water is a good solvent? Or what does water being a good solvent allow your body to do? Transport. Okay? You have a circulatory system. You absorb nutrients from your small intestine into your circulatory system. And then they're transported around through arteries and veins and capillaries so that those nutrients can get to all of your cells. Without water being such a good solvent, we wouldn't have a good way to conveniently move both solid materials and gases around the body from one place to another. Now, there's a downside to water being a good solvent, and that is this transport media can become contaminated by things dissolving in water. And so the transport media can become polluted as well. One of the pollutants that can be released into your body's transport media is produced as a result of the catabolism of proteins. And as proteins are broken down, ammonia can be produced. If ammonia builds up too much in the blood, it can cause serious physiological problems. And so we have to have an excretory system to get rid of that, to convert the ammonia to something like urea, for example, that can then be excreted. So being a transport system is kind of a double-edged sword. It's got benefits, but it can be polluted as well. Well, all of these properties that water has, that life is so dependent upon, come down to one thing, and that is the polar nature of water. And the fact that water molecules, because they are polar, can hydrogen bond with one another. So y'all okay with the idea of covalent bonding and how that differs from ionic bonding? What happens to electrons in covalent bonding? What do two atoms do that are interacting by covalent bonds with those electrons? They're sharing, okay. And if the sharing is equal, like it is in the bonding between two hydrogen atoms to form hydrogen gas, then we call that equal sharing a nonpolar covalent bond. Okay? Water molecules, though, don't equally share electrons. The oxygens tend to be electron hogs. They have a strong attraction for electrons. What's that phenomenon that refers to how well something attracts electrons? Rid of electronegativity. Okay. Oxygen is very electronegative. So it tends to pull those electrons away from the hydrogens. So what happens is the electrons spend a little bit more time around the nucleus of the oxygen than the hydrogen. The electrons being negatively charged give this oxygen a temporary negative charge, and the absence of the electron gives the hydrogen a net positive charge. So this is a polar covalent bond, and it results in these transient charges. What happens if this water molecule is in the vicinity of this water molecule? The transient negative charge of this oxygen is attracted to the transient positive charge of this hydrogen, and we get a weak electrostatic interaction between them. We get a weak attraction. Are hydrogen bonds very strong? No, they can be easily broken, but they reform again. What are a couple of environmental factors that influence whether a hydrogen bond is broken or not? What's a way I could manipulate those water molecules to probably ensure that the hydrogen bonds would be more broken? Heat, okay? I can change the temperature. What's another parameter that I can change that might influence that besides heat? Pressure potentially, although in biological systems we're typically talking about pressures being more or less not changing. Another phenomenon that can influence hydrogen bonding is pH. And we'll talk about that in a little bit. Those are going to be very important concepts, particularly when you do this enzyme lab we're going to do down the road, where we vary some environmental parameters. And one of the parameters that will vary in our experiments is, how does your enzyme respond to changes in temperature? Another parameter we're going to look at is, how does your enzyme respond to changes in pH? Those two particular phenomena are very important because they can influence the nature of hydrogen bonding. And what we're going to see is that hydrogen bonding is an important stabilizing factor in the structure of the biological molecules. And if we change temperature or pH, we can change the structure of a molecule like a protein. You know what we call that change in structure of a protein in response to temperature or pH? Denaturation. Okay? If I take this DNA molecule, remember that the double helix of the DNA molecule is held together by hydrogen bonds between the complementary base pairs, A, T, G, and C. If I take this DNA and I heat it up, what's going to happen to the DNA molecule? Hydrogen bonding is going to be disrupted. And the DNA molecule is going to fall apart. It's going to denature. Biologically, is it significant or important that hydrogen bonds are a little bit strong, but not too strong? As far as DNA is concerned, what's the advantage of hydrogen bonds not being too strong?
It's easy to copy it.
Absolutely. Okay, at some point in time, this molecule is going to have to denature. The two strands of the double helix are going to have to separate so that the two strands could serve as templates for 2 new strands, and that's going to be involved in the process of chromosomal replication, for example. So these are kind of interesting bonds. Individually, they're pretty weak. But collectively, if you have enough of them, they can be strong enough as an aggregate to help to stabilize the structure of important molecules, like proteins or like nucleic acids. And that's why temperature is such an important parameter that organisms have to be able to control, especially if you're a multicellular organism. You don't want your cells heating up too much, or proteins are going to start to denature, nucleic acids are going to start to denature. Already talked about that. Here's another example of cohesion. Water molecules tend to have surface tension. And you can see here that the water molecules almost have kind of an elastic property where a film of water can form on the top of a liquid. And here's an organism called a water strider that has exploited that particular environment and can move along on the surface of the water on the surface tension. We're going to talk about this a little bit more in just a minute, but this whole phenomenon of water being a good solvent is really, really important. When something dissolves in water, what happens is the substance that's dissolving, in this particular case, table salt, sodium chloride, is separating into its ions in the presence of water. And that particular phenomena is called dissociation. So here you can see that the sodium ions and the chlorine ions have been separated. Something's interfering with the normal electrostatic interaction between the positively charged ion and the negatively charged chlorine. And basically what's interfering with that are these water molecules which are surrounding the ions and kind of insulating them from interacting with their negatively charged counterpart. Why are these water molecules dissociating the sodium chloride? Because of the polar nature of water. In this particular case, the positively charged hydrogens right here in white are attracted to the negatively charged chloride. Over here, the negatively charged oxygens are attracted to the positively charged sodiums. So this phenomena of dissociating the sodium chloride into its ions is what we call dissolving. And that sodium chloride dissolves in the water. It's much easier to transport these ions, which are needed for cellular function, in an aqueous environment through blood than it is to trying to move salt crystals throughout the body. Okay? Well, one of the things that we're going to be particularly interested in are compounds called acids. For example, when hydrogen chloride, HCl, is placed in water, it dissociates into hydrogen ions and chloride ions. Okay? In pretty much the same way as the sodium chloride did. Okay? Shells of water molecules surround the hydrogen and the chloride and insulate them from one another, causing those molecules to fall apart or dissociate. By definition, hydrogen chloride is an acid. Why? Because when it dissociates, it releases these hydrogen ions. What's another name for a hydrogen ion? Okay, here's hydrogen up here, right? Atomic number 1. What does that mean to have atomic number 1? 1 proton. And in its elemental state, how many electrons does it have? 1, okay, because it's electric neutral. They balance out. What is a hydrogen ion? It's a hydrogen atom minus an electron, which is what? A proton. So hydrogen ions are also called protons. acids are compounds that when they dissociate in water, release protons. So H2SO4, sulfuric acid, put it in water, it produces protons. Nitric acid produces protons. The more acidic a solution is, the more hydrogen ions it releases during its dissociation. So strong acids produce a lot of protons, weak acids, not as many. Well, if you put sodium hydroxide into water, it also dissociates. Those positively charged sodium ions are attracted by water, as are the negatively charged hydroxyl ions. So sodium hydroxide dissociates as well. But instead of producing protons, it produces hydroxyl ions. Substances, when they dissociate, produce hydroxide ions are called bases. So sodium hydroxide is a base. Oops, got a mistake here. So the more basic solution is, the more hydroxide ions it produces. It's a pretty simple compound. What's interesting, though, is that water molecules also dissociate. They do this spontaneously. They do this on their own. They're not really that stable. And water molecules can spontaneously dissociate into hydroxide ions and protons. They don't do this very readily. If we take a quantity of water and did an analysis of at any given time how many of those water molecules had dissociated, only 1 out of every about 554 million water molecules would have dissociated. It's a pretty low rate, but it still does happen on its own. Okay, so here's the dissociation of water. The hydroxide ions and technically hydronium ions, essentially protons. So, is water an acid? Yes. When it dissociates, it produces protons. Is water a base? Yes. When it dissociates, it produces hydroxide. Molecules that are both acids and bases are said to be amphoteric. Now, where we'll pick up Friday, tomorrow, will be to talk a little bit more about this concept of water dissociation in the context of the phenomenon of pH. Talk a little bit about pH, how the pH scale was derived, and particularly its implication for biological systems. So if you haven't been reading section 2.5, make sure you do that. And then we will move into section 3, and particularly the beginning couple of pages of that chapter, talking about the functional groups that are kind of the building blocks of biological molecules. Okay? So if you have your syllabus thing signed and you want to leave it here, that's fine. Otherwise, I need by Tuesday.
And if you want to take live sections, please let me know by Monday.
Okay. And if you have the seating chart someplace, please drop it up here too.